Hello and welcome. I am absolutely thrilled to have you here today. Let's just dive right in, shall we? Today, we're taking a dynamic, spatial tour of the fundamental building blocks of biology. This explainer isn't just going to be us reading off definitions. No, it's an outside-in journey. We're going to visually unpack the architecture of life, moving layer by layer, barrier by barrier, right into the very core of biological existence. So I want you to start by asking yourself, what does an entire universe look like at literally 1,000th of a millimeter? Imagine shrinking down, completely shedding your normal human scale to explore this bustling, active world that is totally invisible to the naked eye. We're treating the structures we talk about today not just as abstract concepts, but as actual physical places we're visiting on this microscopic journey. Now, every great journey needs a reliable map, right? And our biological glossary is exactly that. Every structure, boundary, and fluid we encounter on our spatial tour today comes directly from this first piece of source text. We're translating these basic written definitions into a dynamic visual expedition, complete with diagrams and motion pictures as we explain things along the way. And later on in our journey, we're going to rely on this second part of our glossary as well. This is where we'll uncover the really vibrant pigments, dynamic processes, and cellular cavities that keep this tiny world running. Between these two foundational texts, we've got the whole shebang. We have absolutely everything we need for our expedition. But to even begin, we need a vehicle. As our source defines it, a microscope is an apparatus used for observing tiny objects. And what's really fascinating is that without this apparatus, this entire world we're about to explore literally wouldn't exist to us. It is the essential key that unlocks the door to the invisible universe, letting us actually perceive these incredibly tiny, complex objects. So, looking through that apparatus, we arrive at our primary destination, the cell. The text defines it perfectly as the smallest structure and functional unit of an organism. I absolutely love this definition because it highlights two key things. First, structure, meaning it has actual physical architecture we can explore. And second, functional, meaning it's alive, it's working, it's the absolute baseline of life. Now, before we zoom all the way in, let's briefly look at how these units actually organize themselves. Notice the distinct difference here. Life can be a solo act, which we call unicellular, an organism consisting of a single cell. Or, like you and me, it can be this massive, coordinated metropolis, multicellular, an organism consisting of many cells. But here's the crazy part. Whether it's one or a trillion, the fundamental architecture inside remains incredibly similar. All right, let's calibrate our scale. We're shrinking down to the level of a micron, which is precisely 1,000th of a millimeter. Just think about that for a second. We're taking a millimeter, which is already a tiny little mark on a ruler, and slicing it into 1,000 equal pieces. That is the impossibly small, incredibly dense environment we are now entering. Section 1. The outer boundaries breaching the cell wall. Boom, the first thing we hit is the cell wall. As defined here, it's a rigid layer outside the plasma membrane of the cells. You can really see in the dynamic diagram how this acts as physical armor. It's tough, it's structured, and it gives the cell its definitive shape, while protecting the delicate little ecosystem inside from the chaotic environment outside. Moving just past that rigid armor, we encounter the plasma membrane. Our source defines this as a semi-permeable membrane, which bounds a cell or surrounds the protoplasm. So the really crucial point here is that while the cell wall provides structure, this membrane is the true bounding envelope. It's this flexible skin holding all the inner, living contents safely together. But this skin isn't just some sealed bag. It's a highly intelligent gatekeeper. The keyword we just saw was semi-permeable. If you watch the selective motion in the graphic, it acts kind of like a microscopic bouncer, dictating exactly what is permitted to come inside with us and what gets locked out. It only allows movement of some liquids and gases to pass through it. Section 2. The living fluid swimming through the cytoplasm. Okay, thanks to that semi-permeable gatekeeper, we've been granted access. We're now completely immersed in the protoplasm. According to our glossary, this is the colorless material comprising the living part of a cell. Notice that it's kind of an umbrella term. It actually includes the cytoplasm, the nucleus, and other organelles. Essentially, everything inside the plasma membrane that possesses the spark of life is part of this continuous, dynamic, colorless substance. To get a bit more specific about exactly where we are floating right now, we're in the cytoplasm. This is defined as the living fluid of a cell encircling the nucleus but inside the plasma membrane. It's the physical medium, the ocean, if you will, that fills the space. All the specialized structures of the cell are suspended and functioning right here in this living fluid. And wow, is it a busy place? Just look at this dynamic process happening all around us, endocytosis. Our source describes this as the process wherein materials move into the cell rather than being engulfed by the cell. The motion graphic perfectly illustrates how the membrane elegantly draws materials inward, depositing them directly into the cytoplasm, where we're swimming. It's just this highly coordinated delivery system. Section 3, the control center. Deep dive into the nucleus. Now, what is truly staggering about the nucleus is its sheer complexity. It's a cell organelle containing chromosomes, and it's actually made up of multiple distinct parts: a nuclear membrane, a nuclear reticulum, nucleoplasm, and a nucleolus. It's honestly like a giant Russian doll. We moved inside the cell, and now we find a whole other highly structured compartment nested deep within. Let's zoom in even further, right into the very heart of that Russian doll. Here, we find the nucleolus. Our source concisely defines this as a globular structure present within the nucleus. So, it's a distinct, dense, spherical command node sitting directly inside the already dense nucleus. And finally, here we reach the absolute core of our journey, the blueprints of life itself, the chromosomes. These are defined as the nucleoprotein threads of hereditary traits. Just imagine these delicate, infinitely complex threads packed securely within this inner vault. They carry the instructions for everything the cell is and everything it will eventually become. Section 4. Pigments, energy, and storage. All right, let's zoom back out and walk through this visual summary of the plastids at work. First up, we have chloroplasts containing green chlorophyll, which is absolutely vital for photosynthesis. Then we have colored chromoplasts. These contain carotene, a yellow pigment that assists chlorophyll during photosynthesis, and xanthophyll, an orange pigment assisting in pollination. And finally, we have leucoplasts, which are entirely colorless, and their specific job is to store food. This color-coded diagram really shows just how specialized these microscopic structures are. Continuing our swim through the cytoplasm, we also encounter these large structural spaces called vacuoles. Our source text simply defines them as fluid-filled cavities within the cytoplasm. I like to think of them as dedicated storage tanks or structural reservoirs, just floating around in that living ocean we discussed earlier. And to complete our structural tour, we have to ask, well, what's inside those tanks? That brings us to cell sap. It's precisely defined as the fluid present within the vacuole. So it's a distinct fluid, totally separate from the surrounding cytoplasm, safely walled off inside those cavities to serve the cell's storage and structural needs. We've breached the outer walls, we've swum through the cytoplasm, penetrated the nucleus, and witnessed the colorful engines of energy. So, the really crucial takeaway here is this. If all this dense, functional, magnificent complexity exists packed into a space of just 1,000th of a millimeter, what else is hidden in the microscopic world? Just waiting for us to point our apparatus at it. Thank you so much for joining me on this incredible tour today. Keep wondering, keep exploring, and I'll